Okay, so thank you for joining this session. And what I'll talk about here is nanobread genome editing, GPCRs, and illuminating the function of native receptors. And this is part of the award-winning Pharmacology from Around the World series. So I'd like to start off by sending a big thanks to the BPS International Advisory Group, who extend the invitation to speak here today, as well as ASCEP, who awarded me the Satara New Investigator Award, as well as BPS ASCEP Outstanding Investigator Award in 2020, which has led to this presentation. I'd also like to thank my mentors and postdoctoral supervisors, Professor Steve Hill at the University of Nottingham and Professor Kevin Flieger at the University of Western Australia. Without their support and guidance, none of this work would have been possible. So I've asked not just to talk about research today, so I will start off with that, but also talk about later in the presentation about my experiences with different pharmacology societies, as well as some of the things I've learned along the way. So my research is mainly focused on G protein coupled receptors, or GPCRs for short, and these are a very large family of membrane receptors found widely throughout the body. Now, I'm sure you're aware that GPCRs are a very useful therapeutic target, and currently, if you look at the literature, you'll find that approximately 30% of drugs on the market target a GPCR or related protein. So what we're very interested in, and this has been the main focus of my research, is how GPCR signaling is modulated either through the formation of GPCR dimers or how different cellular contexts modulates GPCR signaling. So really we want to investigate and decipher the impacts of these different factors, as it helps us not only understand how a receptor functions, but gives us the opportunity to really develop new therapeutics. So the main technique that I've used to investigate GPCR function, or at least the molecular pharmacology of GPCR function, is bioluminescent resonance energy transfer, or BRET. And this is a technique I really first came across, started to use when I moved to the University of Western Australia to work with Professor Kevin Flieger. So BRET is the non-radiative transfer of energy between donor-luciferase and fluorescent acceptor when they're in very close proximity. And you can use this biophysical property to investigate things such as GPCR signaling. You can also look at things such as GPCR dimerization. So if you looking at GPCR dimerization, for example, if you have one GPCR with a luciferase on it and the other with a fluorescent acceptor on it, you have these both expressed in the same cell, and these two receptors interact or form a dimer. You get close proximity between the donor luciferase and fluorescent acceptor. You get BRET between the two, which you interpret as a formation of a dimer. So in practice, the development or generation of a BRET-based assay to look at either GPCR signaling or dimerization is relatively simple, particularly if you're using a transfection-based method. Here you would have plasma-based expression vectors expressing your proteins of interest, one fused to a luciferase and one fused to a fluorescent acceptor. As I said, you transfect these into cells and you get expression. You can do your BRET-based assay. However, this is going to lead to high levels of exogenous expression, so you're going to get overexpression of proteins of interest, and this is going to change the normal cellular context. Particularly, it's going to change the normal stoichiometry between proteins. And if we're interested in receptor dimerization and we have to overexpress our proteins of interest, we are running the risk here of forming or causing the formation of GPCR dimers. So ideally, if we're looking at receptor function or molecular pharmacology of receptors, we're using BRET, ideally we'd be using native systems without overexpression. So I suppose about 18 months before I started working with Kevin at the University of Western Australia, and with these BRET-based techniques, the CRISPR-Cas9 genome editing papers were published, and this has really revolutionized a number of fields now, but it has really provided us with an opportunity to start to develop BRET-based assays with natively expressed proteins. So a traditional BRET-based assay would be transfecting in our proteins of interest, getting overexpression. What we can do with CRISPR-Cas9 genome editing is go in and directly then edit the native genome and insert our BRET-based tags directly into the endogenous genome, resulting in cells expressing a native protein with a tag. So here we're not adding in our protein plus our tag, we're just adding in the tag. So if our cells aren't normally expressing our protein of interest, this process isn't causing the expression. So when we started this work, well, I suppose we were a bit nervous. We weren't sure if there was ever going to be enough endogenous expression to do a Bread-based assay. We knew there was going to be very low levels of expression, but we weren't sure how low this was. And this is just an example of how low the endogenous levels of expression are compared to overexpressed cell lines that we use in our Bread-based assays. So this is comparing a null tag, a luciferase tag CXCR4 that's overexpressed in the black bar compared to endogenously expressed CXCR4 with this luciferase tag, either in HEC293 cells or HeLa cells. So these are genome edited lines. And we're getting about 40 to 50-fold lower levels of expression, which is very, very low. So as I said, we're a bit worried that we will never actually be able to detect a Brett signal with these low levels of expression. However, in hindsight, this was probably unfounded. As you can see here, we're going to get some very nice Brett-based data using these genome-edited proteins. Here we're looking at fluorescent ligand binding by nanoBret with genome receptors. In this assay, we have a receptor with a luciferase on it, a fluorescent ligand. If that fluorescent ligand binds the receptor, we get Brett between the donor and acceptor. So you can see here we're getting very nice saturation as well as competition ligand binding results, even though we have very low levels of expression. So that was binding, and that's a relatively straightforward assay because we only have to tag one protein, but we also want to look at how receptors signal when we're using genome-edited proteins. Here we're looking at beta-arrestin recruitment to a receptor. So this is a typical assay that would be done by Brett or a luciferase complementation approach. Here we have a receptor with one fragment of a luciferase and arrestin with the other fragment. We activate the receptor, we get recruitment of arrestin to the receptor. This luciferase recomplements and we get luminescence output. So here what we want to do is use genome editing to decipher what effect changing the relative stoichiometry of either receptor or arrestin had on the kinetic profile of recruitment as well as the pharmacology. So for this, we set up a number of different assay orientations. Here we have a genome edited cell line with CRISPR editor or endogenous beta-arrestin being recruited to overexpress CXCR4. You see it's very stable kinetic profile of arrestin recruitment. But if we change this assay and orientation, so we now have genome edited CXCR4 looking at recruitment of overexpressed arrestin, it's very transient kinetic profile. Again, this is showing us that depending on the relative levels of receptor and arrestin expression, we do see differences in the kinetic and pharmacology of recruitment. So the last graph here is an assay orientation where we have both arrestin and CXCR4 endogenously expressed in one cell type. This is a very pleasing experiment to do, as when we stimulated with CXCR12, this is the agonist for the receptor we're using here, CXCR4, we saw an increase in luminescence. To our knowledge, this is the first demonstration we could observe protein-protein interactions in live cells using this approach. So really the research that led to the ASCEPT Awards is really around overcoming some of the major limitations of Bread-based assays, and that was the requirement for overexpression, how these may confound our interpretation of the results. And what we're starting to decipher now in different assays and in different cell types is we're seeing differences in both the kinetic profiles as well as the pharmacology of different GPCRs. So in the last 5 minutes or so, I'll talk about less about research, but more about my experiences both with pharmacology and how my career's progressed at this point. So over the years, I've been involved in a number of different societies. The main society has been ASCEPT, the Australian Society. I joined as a member in 2008 and been a member ever since. Also a member of BPS from 2017 to 2020 when I was in the UK on a postdoctoral research fellowship. I've also been involved with IUFAR and a number of committees in between 2018 and 2020. So I'll mainly talk about ASCEPT because this is the main society I've been involved with for a large period of time, and they've been very important, I think, to my career progression. They've given me a huge range of opportunities right the way through from undergraduate level up to postdoc, PhD, and through now as an early career researcher. In general, I found ASCEP and really all societies be very supportive of their student members, not only through student prizes and opportunities to present and travel grants, but also different career development activities they do. I think this is really in recognition that if we're going to grow the field of pharmacology and support it, we really need to be developing and supporting the next generation coming through. So I put here, get involved. I think pharmacology societies, like any community, is really are as good as their members. So I think when I say get involved, this is really around if there's events being put on, participate in them. So if there's workshops, symposia, training days, career development days, go to these things. They're really good for your career development, but also really good and easy ways to network with potential collaborators and potential future employers. Also found if you can contribute back to these societies, you're going to get a lot more out of them. And I've been really fortunate, particularly through ASCEP, to start to contribute to some of the things that I got a lot out of as I was coming through as a student. I think the last thing with the pharmacology societies, and I suppose any society in general, is if you want something from them, they're really receptive to hear what you want. So if you think a different society should be doing more of something or less of something or something different, let them know, because 9 times out of 10, you're going to have a very supportive ear and they're going to be very receptive to that feedback. So the next thing I'll talk about is, I suppose, some of my experiences, and I suppose some of my tips on how to develop your career. So I've had to think quite hard about this. And I think my overarching advice is really, you need to work out what you enjoy. You need to work out how the best you work, how the best you learn, and how the best you interact with people. And if you can get this awareness in yourself, it makes everything a lot easier as you go through your career. It's really about understanding your strengths and understanding your weaknesses and working on your weaknesses, but also using your strengths. So apart from that, I think the main thing is mentorship, and that's really key. You want to find a person or people that you can really work easily with, but you also really trust. You want them to support your career progression and give you really good advice. I think you want people that will push you along the way. I don't think you want people that just sit back and say, yeah, that's good, that's good. You want people that push you out of your comfort zone. That said, I think you also want people that will let you take calculated risks. And that's really my second point, which is to take these risks when they come along. A lot of time I think of this from an experimental standpoint, but I think it also applies a little bit to career progression. So sometimes, or a lot of time, you can have these really great ideas where you want to try something new or try an experiment, and you can always find a million different reasons not to do that experiment or not to try something new because it's safer and it's easier. But sometimes I think you just really need to take that leap of faith, and within your career, it's really great when you can reach that point where you have faith in your own experimental work and your ideas and yourself and your training, that you can take that leap of faith and say, I've designed this experiment well, the question's worth asking, let's go ask the question, even though it's risky. So for me, this was developing these CRISPR genome editing techniques for Brett. We had no idea if they were going to work when we started them. They were risky. They took a lot of time and effort to develop, and it could have all fallen over quite badly, but we were confident in our design. We figured that if it did work, it's going to give us some really insightful information. And if it didn't, then at least we know that also. So I think another point here, and this is advice I got very early on, is to find your own niche. Find something you enjoy, develop this niche over time, incorporate those skills into you, make yourself different, be different from everyone else in the field. This is not just around techniques, but this could be just a whole range of complementary expertise that you can apply to new questions. I suppose the thing here is, find your niche. And the last thing here is plan for your future. This is obvious. Obviously, if you don't plan, you're only going to get to where you want to be by luck. I think one of the best ways of planning for your future is look around you, find those people who are 5 to 10 years ahead of you, and work out how they got to where they were. Not just to copy them, but to learn from them, work out what skills they developed, work out what they did to get where they were. I've also put here, be adaptable. The reason I put this here is because plans change, your priorities change, and if you shouldn't feel bad if you put all this effort into pursuing one career pathway and then you decide you want to do something else. I don't think all that effort you put into a different career pathway is ever a bad thing, because 9 out of 10 times those skills you've developed are going to be transferable. So last thing is how the ASCAP award has impacted my work and career progression. Since it was awarded, I'm going to put old mate SARS-CoV-2 up here to say really it's, I was awarded this at the end of last year, so there hasn't been much time since. But I will say that the work I've been doing and these awards and this exposure has really allowed me to develop a number of different collaborations, as well as pursue different questions that I wouldn't have otherwise. So I'd like to thank you all for your attention, and I do like to thank a lot of people. I do need to thank a lot of people who allow this work to progress, who've helped me along the way, particularly my mentors, Kevin and Steve. Again, without their support, none of this would have been possible. There's a whole range of people working very hard in the background here as well that have supported me to these studies. So I'd like to thank them sincerely. And with that, I'd also like to thank ASCAP and BPS for the opportunity to present here. With that, thank you very much.